In this video we will discuss about the UPR response reporter. So for those who don't understand what this UPR is the unfolded protein response that is usually occurred whenever there is abnormality in the endoplasmic reticulum. And one of the common cause for this UPR response is that most of the unfolded protein response or amount of unfolded protein response amount of unfolded proteins increases within the endoplasmic reticulum, or if there is a disruption in the calcium homeostasis that is maintained by the endoplasmic reticulum. So in that case it will trigger the unfolded protein response. The unfolded protein response has three components, or it has three major signaling activation. First one is the ATF6, that is not mentioned here, and next two is the PERK pathway, and another one is the IRE1 pathway. In ATF6 what happens is that the ATF6 that is usually found in the intracellular membrane of the or internal membrane of the endoplasmic reticulum get move into the Golgi apparatus and then cleaved off at a specific site within the Golgi apparatus, and then portion of or part of those cleaved ATF6 will move to the nucleus, where it will activate the pathways that helps in restoring the healthy state within the endoplasmic reticulum, or it will activate the genes that can reduce this UPR response, either by one of the way by which ATF6 does is by upregulating the proteins that are involved in ERAD so that they can take care of the unfolded proteins. At the same time, they will increase the amount genes or proteins that are involved in protein foldings, such as chaperones, intracellular chaperones, calnexin, calreticulum. So this is how ATF6 functions, and similar to ATF6, this PERK and IRE1 also function. So we will understand in this case how this reporter functions. So this is the, we can say, sequence or time. This is the framework of the reporter. So in normal case what happens is that the translation usually occurs in the ORF1 or ORF2. We can see that start codon and the stop codon in the ORF1 and ORF2. However, whenever there is a ER stress, the translation shifts from ORF1 to ORF3 because of the epigenetic changes. And in this case what happens is that the translation begins in this ORF3 part and ends in UAA. And during this region, the reporter includes this M scarlet fluorescence signals. So whenever there is a ER stress, this reporter translation will occur from this side, and simultaneously this M scarlet will also get translated. So in this way, this PERK reporter can act as a signaling indication whenever this UPR response triggers this PERK signaling pathway. And simultaneously, if these IRE1 pathways also get triggers, then this this reporter will also show a green signal, or this reporter will show a green signal. So how this reporter works is that in case of normal condition, this is the XBP1. In case of normal condition, there is a intron that is present in the XBP1 that prevents its translation. So because of this, what happens is that prevents a complete translation, or we can say the translation ends at this site. Whenever there is no ER stress, this intron is not removed. The translation ends at this site. So this green signal will not get translated. However, whenever there is a ER stress, this intron gets removed, and because of this, there is a frame shift occurs, and the final stop codon will not be available at this site, whereas the final stop codon will be available at this site. So whenever there is a ER stress, this intron is removed, and that whole translation will occur till this end. So during this case, this M neon green will also get translated. So we will see a green signal whenever the UPR response leads to IRE1 activation if those cells contain this reporter. And one thing that we need to we also notice that there is an NLS sequence in both PERK reporter and IRE1 reporter. So what this NLS signal does is, NLS signal is a nuclear nucleation nucleus localization signal. So what it does is, whenever this whole reporter get translated, it will move these reporters into the nucleus, so that we will have all these signals concentrated into a small places, or we can say within the nucleus, so that we can get a bright signal. If this NLS sequence is not there, then all this whole several translation of this reporter will be spread across the cytoplasm, and we will not be able to see the bright signal, as all of them are spread across a larger area, compared to a condition where NLS signal is present and all of them are concentrated within the nucleus. So this is how this UPR response reporter works. If you need to understand this functioning in detail, you can go through the reference paper that has been provided in the description. That's all for this video. Thank you for your time.